Good afternoon. Thank you for the very kind introduction. And I'd like to express my appreciation to Dr. Munson and Dr. Barker for this kind invitation to speak at the 17th Annual PEC Symposium 2022. Title of my talk, as you can see, is Endogenous T-Cell Therapy, the New Math in Adoptive Cell Therapy. It'd be instructive to sort of understand how we make T-cells when talking about adoptive cell therapy, and two of these strategies, TIL and TCR CAR T-cell therapy, are well known to many in this audience, but I'll briefly review this. TIL therapy, or tumor-infiltrating lymphocyte therapy, was pioneered by Steve Rosenberg at the NCI surgery branch and involves the isolation of lymphocyte-rich tumors from which the infiltrating lymphocytes are then teased out in vitro and expanded to large numbers for adoptive transfer. CAR T-cell or TCR-T-based therapy was heralded by the discovery of Zilak Escher initially, and then brought into the mainstream by Carl June and Michelle Sadelain. This involves the engineering of expression of a transgene receptor, in this case a chimeric antibody receptor, that redirects specificity of lymphocytes to a specific antigen. These cells are then expanded and given to patients. TIL therapy has been shown to be very effective against melanoma in several studies at the NCI, MD Anderson Cancer Center, Moffitt, Sheba Medical Center, and possibly other solid tumors, although the jury is still out on several of these studies. Antigen specificity is not required in terms of a priori knowledge of the epitope or the antigen being targeted. However, it's believed that the TIL population contains many neoantigen and tumor-associated specific cells. It does require surgical accessibility to TIL-positive tumor and is a product of undefined specificity and uniformity. Because high-dose lymphodepleting conditioning regimen is required, generally comprised of high-dose Cytoxan and fludarabine, it does encumber some serious toxicities. In addition, the use of high-dose IL-2, believed to be required to maintain the persistence of T cells or TIL, which were generated in the presence of high-dose IL-2, can also lead to potentially serious toxicities requiring tertiary care level hospitals. Therefore, it is limited in tumor eligibility and also patients who are eligible for some fairly rigorous treatment. CAR T-cell therapy, as you know, is effective against many B-cell malignancies, not just CD19 positive leukemias and lymphomas, but as you may have heard recently, approval was given for treating BCMA positive multiple myeloma. It's potentially off the shelf because it's an engineered T-cell product, but it's also encumbered by serious toxicities, not just from the preparative regimen of high-dose Cytoxan fludarabine, but also because of the cytokine release syndrome or CRS type toxicities that can be life-threatening, and as well as neurotoxicities, which have led to several deaths. It unfortunately is also limited in terms of tumor eligibility and patient eligibility, but no question has heralded a new era in cell-based therapies. These approaches, however, have met with some obstacles in TIL therapy, and not unexpectedly, because this is a product of undefined specificity, has been delayed because there is yet to be an assay which represents potency in the TIL product. CAR T-cell based therapies have been very successful in treating patients, even those refractory to conventional therapy, and a high tumor burden, leading to successful induction of complete responses in several studies here. However, CAR-based therapies have also been met with high relapse rates, upwards of 100%, and as early as 3 to 6 months after. So perhaps there's a reason to curb your enthusiasm and think differently in approach to cell-based therapies. Several limitations need to be addressed: persistence, antigen loss, and serious toxicities. Persistence may be attributable to tonic exhaustion of the transgene expressing T cells, and then the requirement for high-dose conditioning and high-dose IL-2. Antigen loss because high-dose lymphodepletion depletes the endogenous, potentially tumor-reactive T cells, can lead to an absence of antigen spreading and a very high relapse rate, as we've observed already for some of the CAR-based therapies. And finally, toxicities seen here, severe CRS and neurotoxicities, limited ability of using cell-based therapies in combination with other immune modalities. And finally, the unmet need really is the identification of targets with high immunogenicity. Beyond these two major modalities, there is a dark horse, which we have called endogenous T cell therapy or ETC therapy, requiring the isolation of very low frequency cells from the peripheral blood and then application of enabling technologies to expand, enrich, and infuse these into patients. What is endogenous or ETC therapy? As I said, it requires only peripheral blood as a source of T cells. As a result, the extant circulating population, extremely low frequency, requires some strategy in order to enrich and generate a uniform population of antigen-specific T cells for adoptive transfer. It took us quite some time, up to 2 decades really, to bring this to a treatment modality. This involves 2 enabling technologies. One, a strategy to generate antigen-specific memory type T cells, in this case through ALK21 priming of naive population, and an MEMS-based cell sorting strategy to isolate this rare extant population. This peripheral blood source of T cells provides unique advantages. It becomes a highly defined T cell product with uniform specificity and a memory phenotype, thereby obviating the requirement for addition of depletion or high-dose IL-2. In fact, the persistence of these transfer T cells is what makes them so effective and also gives them potential to be used in the treatment of several different tumor types. By sourcing this from a broad repertoire of naive peripheral blood T cells and eliminating requirement for many of these potential toxicities, one can isolate T cells of a frequency really of less than 1 in a million, which he demonstrated in the Science Immunology paper in 2017. These 2 manuscripts central memory T cells can be isolated and expanded to numbers of 10 billion per meter squared with a uniformity of greater than 80%. So the issue of persistence we like to address by understanding what metrics. One is duration and one is the half-life of these transferred T cell clones. In 2012, we used the central memory type T cells to treat patients with refractory metastatic cancer and found that, for example, in this patient with a tumor hanging off his transverse colon that's seen here on the CT PET scan, we were able to achieve a complete response. And accompanying this response was what we observed to be long-term in vivo persistence of a transfer population of antigen-specific peripheral blood-derived endogenous T cells. These MART1-specific T cells, seen here at a relatively high frequency of 5%, 4 months after infusion, persisted at still measurably high levels almost a year. And now 5 years later, we can still detect these circulating antigen-specific T cells in a patient who we can safely say has been cured of his metastatic disease. Whereas in patients with progressive disease, we do not observe such persistence, and patients seem to disappear relatively quickly. We also found, when we examined the surface expression of the antigen-specific population in the T cell product, is that while they go in looking very similar as an effector population, RO positive, CD28 negative, CD127 low, the cells that we were able to detect in the patient's progressive disease retain this effector phenotype, whereas those who achieved a complete response actually reverted to a central memory phenotype characterized by CD28++ expression and very high levels of IL-7 receptor in the circulating antigen-specific population. How do we generate these central memory type T cells? Well, first of all, one must first recognize that surface marker is not enough. Although all T cells are equal, some are more equal than others. And by good luck, when I worked at Fred Hutchinson's, Almo Genetics just cloned a new gamma chain receptor cytokine, which is IL-21, and although it belongs to the same family of gamma chain receptor cytokines, such as IL-2, IL-7, had a completely different property when it was given to in vitro primed naive T cells. This is a relatively simple assay where we took some autologous dendritic cells, pulsed them with the immunogenic peptide, and then assayed for the phenotype and function of these transfers of peripheral blood-derived CD8 positive naive cells. And we found that with IL-21, there was a fairly significant increase in the population of CD27, CD28 positive cells, as well as those expressing CD60 ligand and CD127 compared to those T cells that were primed in the absence of IL-21 or in the presence of one of these other gamma chain receptor cytokines. MFI analysis demonstrates a clear difference between those that were treated with IL-21 during priming and those that were not. Furthermore, this expression of CD28 on the surface of these in vitro prime antigen-specific T cells that were exposed to IL-21 shows sustained expression of CD28 in the fetal population. Functionally, these IL-21 prime T cells also demonstrate increased proliferative capacity, as seen here in the CFSC dilution assay, compared to those which were not treated with IL-21, and that the CD28 expression on the surface of IL-21 was in fact also functional and delivered an IL-2 signal, seen here, when these T cells were treated with a B7 positive target. You see autocrine antigen-driven levels of IL-2, which can be blocked with CTLA-4 Ig when it masks the CD28 counter receptor B7. This was also published in 2005. Confirmatory studies were performed in 2008, and then finally, these IL-21 prime T cells represent a central memory helper-independent CTL, which leads to polycytokine and in fact IL-2 antigen-driven production. These helper-independent T cells belong somewhere in this differentiation pathway with high proliferative capacity and inducible cytolytic capacity. How does IL-21 do this? In contrast to its other gamma chain receptor cytokines, IL-21 signals predominantly through the STAT3 receptor and affects memory differentiation. In a number of studies going back from 2005 to more recently 2020, we sought to understand whether CD28 regulation was STAT3 dependent. And in fact, as it turns out, exposure of naive T cells to signal 1 to its T cell receptor, as well as IL-21, leads to transcriptional upregulation of CD28, which can be blocked with shRNA to STAT3. Since we didn't really have a knockout human model, we were fortunate to find that patients were available through a pediatric collaborator who had Job syndrome. Patients with Job syndrome are functionally autosomal dominant STAT3 negative due to mutation in the STAT3 locus. And these patients, when exposed to IL-21, do not upregulate CD28, as you can see here at the transcriptional level. We found that phospho-STAT3 by ChIP-seq analysis also binds to the distal and proximal regions of the CD28 gene, and that this leads to angenetically more robust phenotype on seahorse assay shown here. Perhaps the most definitive study of the in vivo effect of IL-21 prime T cells was published in patients we treated with WT1 specific T cells. These patients with high-risk AML usually bridge to therapy with allogeneic stem cell transplant, but they almost uniformly eventually relapse. Post-transplant, we gave these patients donor-derived WT1 specific T cells. In one group of patients, they received WT1 specific T cells that had not been primed in the presence of IL-21, so generated with the conventional approach. And you can see in this patient, for example, that the T cell product has low levels of CD27, CD28, and other memory markers, and that the antigen-specific T cells we could detect in vivo after adoptive transfer by day 6 showed no upregulation of these central markers. By contrast, in these two patients, for example, who were treated with WT1 who had T cells generated in the presence of IL-21, we see that the T cell product here have relatively high levels of CD27, CD28. Here, patient 24 as well, and that when we evaluate the antigen-specific tetramer positive population in vivo after adoptive transfer, they have recovered or actually increased the fraction of these central memory type T cells in the peripheral blood. You can see here with high levels CD27, CD28, and CD127. More importantly, though, the patients that received WT1 specific T cells without IL-21 showed a modest persistent levels which were not sustained and required repeated infusions. And unfortunately, all these patients eventually relapsed. You can see here the red line showing the percent leukemia cells, the leukemic blast, reaching 100% of the bone marrow and peripheral blood. These patients unfortunately passing a result of the recurrence of the leukemia after transplant, and even with the treatment of these WT1 specific T cells. By contrast, the patients who received IL-21 prime T cells showed sustained persistence of these transferred T cells. As you can see here, relatively high levels. This is a log scale. This levels up to 10% or more after adoptive transfer and reconstitution of their baseline lymphocyte population, and also no recurrence at all of the high-risk leukemic clone. So the question then is, why would we only use this approach in ETC therapy? Unfortunately, the population of TIL cells and CAR T cells, at least in the manner in which they're manufactured now, gives us largely a population of effector memory and terminal effector cells. So we asked the question, could we treat these TIL and effector CAR T and TCR T cells with IL-21 and recapitulate what we saw with ETC therapy, where we could start with the naive T cell population? Although we see upregulation of phospho-STAT3 in both the naive population, which we showed you previously, as well as in the effector TIL and CAR T population, you see here increase in phospho-STAT3. In patients, effector T cells, exposure to IL-21 does not upregulate CD28, and you see no shift here and no change in the MFI. We believe what's happening is that the naive population, the promoter region of CD28 is exposed and allows for phospho-STAT3 binding, while as T cells differentiate, the chromatin becomes more closed, preventing phospho-STAT3 immediate regulation of CD28 and other essential memory markers. So we reasoned that if we could pre-treat these effector cells with something that would release the chromatin and allow exposure of these sites, perhaps with a HDAC inhibitor, one could reprogram these effector T cells. And that, fortunately, exactly what happened. Pre-exposure to SAHA for some days led to upregulation of the STAT3 signal, when ChIP-seq analysis shows binding to both the distal and proximal regions, which now exposed in these effector T cells. Not only that, but we saw an increase in both CD28, CD62 ligand, granzyme B, and perforin levels in HDAC-treated TIL and CAR T cell populations. You can see here in the TIL population, increase in the CD62 ligand, CD28 TIL positive cells, and that functionally exposure with HDAC, which can be toxic at certain treatment regimens, did not affect killing or expansion, but did continue to maintain the enhanced proliferative response to gamma chain receptor cytokines. We feel, therefore, that this type of pre-exposure with HDAC and then a subsequent treatment with IL-21 can lead to reprogramming of these TIL and CAR T cell therapy, giving rise to a sensory memory type population. This first obstacle, or first limitation of CAR T cell and TCR-T based therapies may be addressed with the use of highly persistent T cells, that we've shown you with our ATC model. But the question of antigen loss and the ability to remove high-dose lymphodepletion because we have memory cells and don't require high-dose conditioning has yet to be addressed. And we believe that the role of antigen spreading is important in the efficacy of cell-based therapies. In this case, a T cell which recognizes its target leads to release of tumor-associated proteins, which then taken up by professional antigen-presenting cells, such as the dendritic cell, and then presents other non-targeted tumor-associated antigens to the endogenous, potentially tumor-reactive T cell population, and an expansion of specificity across multiple tumor antigens. This is what we call antigen spreading. However, if you give patients high-dose lymphodepletion like high-dose Cytoxan and fludarabine, you eliminate the presence of these potentially tumor-reactive T cells, and therefore abrogate any potential for antigen spreading to occur. In 2008, a study we did many years ago using adoptively transferred CD4 positive T cell clones against NY-ESO-1, which we generated at that time by brute force cloning since we didn't have a cell sorter available at the time. We demonstrated that in some patients we will achieve a durable complete response by the transfer of NY-ESO specific CD4 positive T cell clones alone, without lymphodepletion or without IL-2. But what was intriguing to us was that even though NY-ESO-1 is heterogeneously expressed, and T cell clones only targeted specifically NY-ESO-1, we did not see the development of antigen loss variants in these patients who achieved a complete response, and we attributed this to antigen spreading. And to prove this, in fact, we showed that the peripheral blood of these patients who were treated with CD4 T cells against NY-ESO-1 could elicit MAGE-A3, MART1 responses, which we did not target, and that this was probably a surrogate marker for antigen spreading. Subsequently, in a trial that was published a few years ago, we use these IL-21 prime CD8 T cells now, which were HELP independent, in combination with immune checkpoint blockade for patients who had refractory metastatic melanoma. These were antigen-specific T cells targeting GP100 tyrosinase or MART1, a dose of about 10 billion per meter squared, a population that was 80% specific in uniformity. And to this, we added ipilimumab at a dose of 3 mg per kg, starting the day after adoptive transfer. Now, recall that without lymphodepletion or high-dose IL-2 and the use of EDC-based therapies, we were able to overlay the treatment of these patients almost immediately after infusion, because of the absence of toxicities, with another immune modulator, in this case, ipilimumab. Now, the reason you use CTLA-4 is because it provides untethers CTLA-4 from binding to B7 and allows the unfettered ligation of B7 with the high levels of CD28, which was expressed on the T cells. This study, we showed in one patient that had failed both monotherapy with adoptive transfer T cells, as well as monotherapy with ipilimumab alone. The combination of the IL-21 prime T cells with CTLA-4 led to near durable complete responses, seen as far as 5 years from adoptive transfer. Large bulky tumors in this CT scan, which had encroached upon its major vessels, was completely eliminated with only a single infusion of IL-21 prime MART1 specific T cells in a single course of ipilimumab, without high-dose lymphodepletion, without high-dose IL-2, and targeting only a single epitope. In a larger cohort of patients, we saw, as you can see in the spider plot, 2 complete responses, 2 durable complete, and 2 other patients with durable partial responses, and several patients with stabilization of disease, showing that this approach is efficacious, but more importantly, is that this likely led to the phenomenon of antigen spreading. In this chart of one of the patients here, along the z-axis, you see time from 0 to 180 days after adoptive transfer. Along the y-axis is the response as measured by ELISpot assay to interferon gamma. Along the x-axis here, all the different pooled overlapping peptides we use to assay for response to MART1, NY-ESO, GP100 tyrosinase, and MAGE-A3. Although we were just targeting MART1 with our MART1 specific T cells, and you can see here when the T cells were given in combination with CTLA-4, we saw an increase, as expected, of MART1 specific responses. But what was also very intriguing was we saw increase in the non-targeted T cell response to NY-ESO, GP1 tyrosinase, and MAGE-A3, even though we had only treated a patient with MART1 specific T cells, inducing levels of response to these antigens to frequencies as high or higher than that of the transferred T cell response. Pretty dramatic antigen spreading, we believe. And when you flatten this to a TD curve, this is what you see. Same thing that I showed you in the previous slide. except now we represented these responses in terms of the height of individual antigen-specific responses by the different colors shown here, and you can see antigen spreading in this patient following T cells plus 4 doses of ipilimumab, clearly much greater than what we've seen in previous treatments or with monotherapy alone. And then in the 6 other patients who had either stable disease or partial response or complete response, we see increase in antigen spreading phenomenon to these other non-targeted specificities. This patient in particular is quite interesting, patient 7. This patient developed colitis, immune-related colitis as a result of the ipilimumab, not unexpected. But he actually continued to develop a sustained complete response in spite of exposure to high-dose steroids to treat his colitis. And functionally, you see a decrease in the level of activity against these antigens, but these cells were still present, as we saw in the structural analysis, at relatively high frequency. So when the steroids were lifted, this patient was able to maintain a continued functional response against his tumor. And in non-response, we did not see evidence of antigen spreading, suggesting that antigen spreading does correlate possibly with a clinical response. The third problem really here is toxicities, and I think I've addressed this briefly by showing you several studies in which patients were able to receive ETC-based therapies without high-dose conditioning or high-dose IL-2, and yet we continue to see persistence and in some cases significant efficacy. So this approach of ETC-based therapies, we then extended beyond what we saw initially in patients who received CD4 T cells to other cancer types, not just leukemia or melanoma, but also to Merkel cell cancer, sarcoma, mixed round cell liposarcoma. And together with this, have combined this with additional memory programming strategies in vitro to enhance the in vivo persistence of transfer T cells. And what we see from all this basically is that persistence, antigen spreading can lead to durable complete responses, especially when given in combination with other immune modulators. So we've advanced beyond just CTLA-4 to other checkpoint inhibitors, anti-CD137 agonists, the vaccine, and other approaches for other cancers as well. This is the math I've been trying to espouse for some time, and really what we're trying to show here is that using a T cell receptor repertoire that's relatively broad, plus IL-21 priming, gives rise to a population of memory T cells, allowing for persistence of these transferred cells. And this persistence eliminates the requirement for conditioning IL-2. Without the requirement for conditioning IL-2, one can induce antigen spreading and allow for the use of combined immunomodulatory approaches. Persistence with antigen spreading combination therapy, therefore, equates to a clinical response, durable as we hope. The final sort of missing component of all this, the major unmet need really is that a lot of the targets, especially in solid tumors, are relatively limited, or they're not defined in the case of TIL-based therapy, and a robust antigen discovery pipeline is needed. I'll just touch very briefly on this for the sake of time. What we do know is for CAR T cell-based therapies, we have CD19 and now BCMA, and plus a lot of other targets, but this really reveals only the peel of this apple, which is the tumor antigen universe. TCR targets, however, which is the bulk of the core of the apple, a much more diverse and broad repertoire because TCR targets are peptides or bits of the protein processed and presented internally and then brought to the surface in the context of MHC. And so really you can choose from any target, from any intracellular, intranuclear, or even surface source of proteins. Transcription factor can be munched up and brought to the surface. Peptide context of MHC. A lot of these CT antigens, as you see here, are presented as immunogenic peptides and epitopes, but really only a limited number of these have been shown to be immunogenic and capable of inducing a tumor rejection. And the expression of these antigens are present actually at relatively low levels, less than 2 transcripts per megabase overall, and taken into the full panel of tumor types and HLA alleles, what we know is limited to only less than 3% of all patients. So there really is a significant gap here. And the conventional strategy of identifying these antigens has been through prediction algorithms or reverse immunology or use of overlapping peptides, but it doesn't really address the question is, are these peptide epitopes immunogenic? Is it presented? Does it elicit a T cell response? Is a T cell that's elicited of sufficient affinity to recognize these tumor cells? And these tumor cells present the peptide image to sufficient density on the surface to allow for tumor antigen recognition. And this, as I showed you before, really, when you look across the TCGA database, these 4 or 5, maybe handful of tumor-associated antigens, some of which are tissue-specific, some of which are CT antigens, really represent a tiny slice of the entire population of tumor tissues. But we decided actually we go about this in a more rigorous manner, go where the money is, loot off the peptide from the tumor cell here, subject it to LC and tandem mass spec, pass it through a number of algorithms. So we know that peptide is absolutely processed and presented, otherwise we wouldn't find it from the eluted MHC. And then not only do we define this peptide epitope repertoire, but we also demonstrate empirically using the ETC workflow, because the ETC workflow allows us to isolate very rare tumor-active T cells from peripheral blood to generate T cells and also validate the immunogenicity by showing that these T cells recognize endogenous presented tumor-associated antigens and epitopes. And that furthermore, if we take the alpha and beta chain of T cell receptors, we can show killing of their respective targets. And using this, we have a huge number of epitopes which are well defined. And you can see here both tissue specific in the case of prostate cancer, as well as more tumor associated antigen specific epitopes representing more than 75% of all HLA alleles globally. And not only are these found in a number of tumors, also present in very high levels. Not only can this peptidome be used for generating ETC and TCR-T-based therapies, but also lead to immunodiagnostics in determining antigen spreading and T-cell correlatives. I'm going to end there and thank all the people who did the work, as many of them as I can here, Kei Pan and Yulin Chu for the immunopeptidome, Ivy Lai and her team of cell therapists for several of these seminal studies, Dr. Wang for the auditorium primary studies, Xiao, who is master of all trades, responsible for several of these studies. So here, certainly new and prospective studies across a number of collaborators here, Dr. Rai, Dr. Maitra, Dr. Lai, in the Rare Tumor Initiative, and also the SynBio and gene editing team at Rice University. And then some ongoing collaborations, which I haven't had time to talk about, but I think funding was indispensable from the Park Institute, CPRIT, Stand Up to Cancer in one of the Dream Team Immunology initiatives, as well as from the NIH and the Cancer Research Institute. Thank you so much for your time, and I'll take some questions live.